From Microbe TV, this is TWiP, This Week in Parasitism, episode 244. I'm Vincent Racaniello, and you're listening to the podcast all about parasites. Joining me today from Fort Lee, New Jersey, Dixon Despomier.
Hello, Vincent, and everybody else. We usually give a brief weather report now. It's sunny outside, moderate temperatures, low humidity, perfect weather. I'll take 120 days of this every year, but that's not going to happen.
Also joining us from South Africa, Christina Naula.
Hello, not my usual spot to sit. My weather report, as you can see from my hair, it's really windy, but other than that, it's really pleasant. And I'm attending a conference here on One Health, so perfect.
You know, your internet is better than at home. I hate to say.
Yes, I know. I was really worried it might be rubbish, but actually, it's better here than at home, isn't it?
It's in a hotel, or is it a house or something?
Yeah. No, it's a hotel. Yeah, my Glasgow internet is appalling.
We have a guest today. We're all over the world today. We actually have someone from Glasgow to fill in the Glasgow position. She's a professor at the University of Glasgow. Lilach Sheiner, welcome to TWiP.
Thank you. Thanks for having me. I wish I can have a great weather forecast for you, but because it's almost 11 at night here, it's basically just dark and a bit cold, which is actually what Glasgow is usually like.
Miss Christina's always talking about cold and windy.
Yes.
But of course,
and it always feels like February.
And
sounds like the opening to a mystery novel. It was a dark and stormy night.
This is how our story begins.
This is how our story begins.
So before we get into science, I'd like to remind everyone that if you enjoy these science programs, we'd love to have your support. The production company Microbe TV is a nonprofit entity in the U.S., and so your contributions are tax deductible. We exist on your donations, on your love for our work. So go to microbe.tv/contribute. We have several ways that you can help to bring science to everyone, because we want everyone to appreciate how amazing all the sciences are. So Lilach, we're going to talk, what a lovely name, like a flower, right?
Thank you. Yes.
We're going to talk about an amazing paper that you were an author on today. But before we do that, tell us a little bit about your history, where you're from, your education, and how you got to where you are, so forth.
So I'm Israeli, born and raised, was there until kind of my very early 20s. So that's where I also studied my undergrad and also my master's degree. I fell in love with microbes thanks to a university professor who taught me microbiology. And the one thing I like to remember, her name is Shula Michaeli, and one thing I like to remember is she was speaking to us about microbes, and it felt like she was seeing them as she was explaining how they behave. Very inspiring. And so I did my master's program with her, and when I finished, I asked for her advice. You know, this might be an advice for all young or early career people. If you're not sure what you're doing next, ask somebody you really like what they would have done if they were in your career stage. So that's what I asked her, and she told me about this person who just cracked genetic manipulation for Toxoplasma gondii, which is Dominique Soldati, and that's the person I went to do my PhD with. So she taught me how to genetically manipulate Toxoplasma gondii, and that's what I've been doing since then. I've done my postdoc in the US and started my own research group in Glasgow 10 years ago exactly.
All right. So the paper today is all about Toxoplasma. So maybe before we get into it, you could give us a little background on the life cycle.
Sure. I think Toxoplasma is often for those people who heard about this parasite, it's often the cat parasite or the zombie parasite, cat zombie. And that comes from the fact that the sexual reproduction of the parasite takes place in a cat. And then the cat feces would release oocysts that are infectious in the environment, and then those would infect what we call intermediate hosts. We call them intermediate because you have asexual reproduction in them. That means that the form of the parasite you get there just keeps replicating itself. It doesn't change anymore because there is not another type that will mate with it and allow some changes. And Toxoplasma is an extremely, extremely successful parasite because it infects a whole variety of intermediate hosts, different types of warm-blooded animals. That kind of life form can infect between intermediate hosts. So, for example, if a person ate a tissue of an infected sheep, that person can get also the asexual form. So that's why people call it the cat parasite. There's a lot of debate in the field, but there's been some evidence suggesting that infection can cause behavioral change, and that's where the zombie part of the story comes from. This is, there's a lot of observation and correlation behind that, but the mechanism is very, very debated. And then the other maybe thing to know about that is the asexual life cycle includes a form of the parasite that replicates very fast and that causes tissue damage, and another form that is dormant, that when the immune system reacts to the parasite, some parasites can go what we call dormant. They're not really dormant, but they're far less active. They're called bradyzoid, and they stick around presumably for the lifetime of the organism. And so in the case of human, these kind of semi-dormant form can reactivate, and that's part of the issue with Toxoplasma. And then maybe the last thing to know about it is that this parasite can cross the blood-brain barrier, which is actually normally there to protect the brain from things that shouldn't get there. But over evolution, Toxoplasma evolved a way to do that, and therefore there is the issue that you get brain cysts of Toxoplasma.
Do we know how the parasite invades the brain?
There's a few mechanisms that are proposed for that. One of them is that it actively, so Toxoplasma is really cool in the way that it moves around from its own kind of actomyosin motor. It has its own way of moving itself forward, and it invades cells also in a very active way. And so one of the suggestions is that it invades some cells and then come out the other side of the blood-brain barrier. There's some suggestion that it hijacks inside cells of the immune system and kind of infiltrates like that. So there's a few different mechanisms that are proposed for that.
Thank you.
So the paper is Nature Microbiology, Engineering Toxoplasma gondii Secretion Systems for Intracellular Delivery of Multiple Large Therapeutic Proteins to Neurons. And there are a lot of authors here. Can you highlight some of the key authors for us, Lilach?
Yes. So the author I'd like to first give a lot of credit to is the first author there, Shachar Bracha. Shachar, and maybe I should also speak about one of the other last author, Oded Rechavi. Shachar and Oded have been struggling with the idea of how do we deliver brain therapy. There's several different brain conditions where we know what the therapy is, we know what the protein that is missing is, but delivering it through the blood-brain barrier has been the challenge for the medical field. And they had this idea of, well, why don't we use a microbe that naturally crosses the blood-brain barrier anyway? And a bit of a fun fact or a fun story. 10 years ago, when I just finished my postdoctoral training and was about to start my lab, I was traveling the United States because I did my postdoc in the US. And before leaving, I wanted to see a lot of the beautiful country. There's so many different landscapes to see. Because we were seeing a lot of the national parks, we have stopped in Vegas for a couple of days. It was just on the way between some parks. And Oded and Shachar called me. Back then it was via Skype. To speak to me about this crazy idea. And we were saying if there is a place for us to start this mad idea of using a brain parasite to deliver brain therapy, it's in Vegas. And so that's when the kind of collaboration between us emerged. And Shachar came to visit my lab the same kind of first few months when I opened it in Glasgow. And she's been a force of nature to drive this paper that took about 10 years to come out, all through those 10 years, sometimes in my lab, sometimes in her home lab in Israel. And also after she graduated her PhD and was doing her postdoc at MIT, which is where she is right now. So definitely those two people I like to highlight. There's many authors on the paper because we've used so many complementary systems there to test the system in different ways.
So, the question I would ask you at the onset before we do anything else is, don't you worry about putting this parasite in someone's brain for therapy?
Definitely. So that's why the Vegas, right? It was a crazy idea back then. And I think it's important to tell anyone who listens to this or reads the paper or think about taking it forward, it's still a very crazy idea. So the important thing is the paper provides a proof of principle, and I'll speak about the details. A proof of principle that we can take the parasites, make them express those therapeutic proteins, make them spit them and put them in the right place. But we're still talking about pathogens. And so a lot of the kind of future work, if we're going to take this and turn this into a real medical tool at any time in the future, which is far in the future, we're going to need to make the parasites less virulent, which the lab is working on at this point, as well as try to control a little bit where they go, which at the moment the whole Toxoplasma field doesn't understand that very well. So there's a lot of work still to do there.
Well, sometimes genius comes out of a crazy idea, so you have to be persistent.
I need to follow up on that, Vincent. You almost took the words out of my mouth. Albert Einstein was once quoted as saying that if your idea isn't crazy, it's probably not worth doing.
That's right.
I really hope he was wrong, because there's also research in my lab that is not terribly crazy, but I still think it's quite cool.
Well, everything looks crazy to begin with, right? And then the truth comes out and it's, we take that for granted now.
Exactly.
So, Lilach, can you tell us some of the problems with protein therapeutics that maybe this system overcomes?
Yeah. So I kind of alluded to it at a very kind of superficial way. The blood-brain barrier is there to protect or to block large molecules from reaching the brain, for obvious reasons. And some of this means that molecules of a certain size cannot reach the brain. Certain hydrophobicity, so whether or not they can cross membranes, and kind of other sort of biochemical features. So one of the ways that molecules can reach there is for example, being synthesized on location. And one of the cool things about Toxoplasma is it's also a eukaryotic cell. So in that sense, similar to cells of mammals and of people. And therefore, if Toxoplasma makes a protein, it will be the right size and the right biochemical properties, most likely, or more so than in other systems, than the way it's being produced in if it was produced in the human cell. So what we did here is we took an organism that can make the protein after it crosses the blood-brain barrier. So all of these issues of size and of biochemical properties, we kind of overcome because we make the protein after crossing the blood-brain barrier. I should also say there was another issue. It's not only about crossing the blood-brain barrier, so that was one issue. The other issue is getting to the right location inside neurons. And that's another thing. Toxoplasma has this system where it can spit proteins into cells. It doesn't even have to be the cell that it actually resides in. It goes around and interacts with a few cells and spit proteins into them. And then those proteins go into locations inside of those cells. So two kind of barriers. Firstly, crossing the blood-brain barrier and then getting into the neuron cells.
Are those the rope trees by any chance that are doing that?
Exactly. So the parasite have three types of secretion systems, right. And we've capitalized on two of them. And you're right, the rock trees are the system that they use to just spit cells into different, spit proteins into different cells. And then the second system is the dense granules, and some of the dense granule proteins get secreted after the parasite is already intracellular.
What's the normal function of the ruptures? We say like they're spitting proteins out, but what kind of proteins? They must have a purpose, really.
Yes. I think a couple of examples maybe is that some of the rupture proteins enters the cell, go into the cell nucleus and induce change in gene expression that, for want of a better word, make the cell a better place for Toxoplasma to reside in, maybe have access to some nutrients or slightly less hostile environment in that cell.
Thank you.
So you had to design fusions for each of these systems, right? For the rope trees and for the dense granules. Give us an overview of how you did that, please.
So this is again a salute to Shachar, who did two, among the many stuff that she's done here, she did two kind of huge undertakings. The first one was to go through the literature of already known brain conditions where the therapeutic proteins was known, and look at all of those proteins and ask which one of those are likely to be expressed in the parasite and are simple enough so that they don't need to have a lot of post-translational modification, and so it's most likely that they'll get expressed and delivered into the right place. And then after doing that, she selected a few tens of those and tried to genetically engineer the fusion between the rupture protein and those therapeutic proteins, or the dense granule and those therapeutic proteins. And then she tested all of those parasite lines that express those fusion and asked, do they express our protein of interest? Is it the right size, indicating that it's the one we wanted? Is it going to the right place inside the parasite, in the ruptures or in the dense granules? Does it really get secreted? Where does it go to in the host cell? So she's done kind of those screens until we came up with the three that are that have been successful in the paper.
I mean, are you looking at diseases like Alzheimer's or other related diseases that have I went blank on the lack of a protein that the prions that get out of control, because we have our own prions, but obviously they're not out of control. Is this something that you want to interact with to try to prevent?
So Alzheimer's is definitely one of the ones we were considering as a future potential condition to target. The one that has been kind of the most, the one we've had the most progress with of the proof of principle in this paper is Rett syndrome. And for Rett syndrome, we were able to express the protein that is missing, which is a transcription factor called MeCP2. And that's the one where we showed both expression, delivery in the parasite, delivery into different types of host cells, and some indirect evidence of functionality.
Right.
So these early experiments where you are targeting heterologous proteins, what you use a cell in culture for these? Is that correct?
Yeah, the very early one we used fibroblasts, so a bit like skin cells, which we culture in tissue culture in culture dishes, and then we infect them with Toxoplasma, and we can do some biochemistry, so collect proteins and see how they migrate on a Western blot, as well as do some immunofluorescence and ask where are they in the parasite and where are they in the host cell. Okay. So yeah, all of those first experiments were done like that.
And you even show that you can use both systems at the same time, right? Both the rhabdoids and the granules, right? Yeah.
That was one of the many. You know how a scientific paper goes through iteration, where expert reviewers would ask you, "Well, why don't you show us this and that and the other before we get convinced?" This was one of the many iterations where reviewers were going, "Oh, well, if you want to tell us that this is a good tool to do something with, why don't you show us that you can express something from both systems?" And then Shachar made that happen as well. I think the rationale there was, perhaps we can use this system for other things. So can we use it as an experimental tool to infect animals in culture, or even organoids or tissue cultures, and bring two different tools at the same time. And yeah, we did that in the paper as a proof of principle.
The beauty of this parasite, as I always used to say to our medical students, was that it knows no barriers. It has no host preferences, and it has no cellular preferences. Yes. So you can virtually work on any system.
Yeah, almost. It infects, the kind of textbook is it infects all warm-blooded animals and all nucleated cells. So very, very broad.
Exactly. I almost add, but we don't include things like echidnas and platypuses.
Yes.
I was actually just going to say, in your paper, and that's maybe something for our listeners to go and have a look. The paper is open access, so some of the immunofluorescence images are absolutely stunning. You can so very clearly see, where in the parasite the proteins are located and where they come from and where you go to. So really, if our listeners would like to have a look, I would really recommend that. It's a difficult paper to read, I won't lie. It took me some time, but it's worth for just looking at the images. That was just admirable, really.
So Lilach, then you move to neuronal cells. Tell us about those experiments.
Yeah. So I have to probably admit I can't tell you a ton about those experiments, apart from the fact that they're quite difficult. So primary neurons don't grow as well for if you're in the habit of culturing cells in cell culture, oftentimes you're used to looking down the microscope and seeing healthy cells, plenty of them, and then you get enough to do your biochemistry, or you get beautiful cells to do your immunofluorescence. Primary neurons are a little more finicky and tricky to grow, and so these were tough experiments to optimize, to make sure that the cells remain adherent and large enough, and especially they become all unhappy when you infect them with the parasite. So also getting the infection rates such that you still have enough adherent cells so you can still capture some immunofluorescence. But those were successful again.
So it was very interesting. You took a protein from neurons that was missing in the neurons, and then you replaced it with the toxo and showed that it worked.
Yeah,
right?
exactly.
That's very nice. And then you moved to brain organoids, right?
Yes.
Where you have a little bit of organization and did the same experiment, right?
Exactly. That again came from some reviewer comments and a new collaboration that joined. So over the 10 years, you can see that there is lots of labs involved in it. So over the 10 years, we kept adding collaborators on. So the Italian groups are the one that did the organoid work. And there we had kind of two advantages. The first one is that, we were able to show also when we're looking at kind of a 3D system with a little more complexity that is slightly more close to the organ. We still get infection, we still get delivery of the protein. And the other thing is that that group had good experience in looking at gene expression patterns and seeing when we bring the parasite and it delivers MeCP2, we can look at the gene expression that results from that and suggest that there is a real functionality or indirect evidence for a functionality of the protein that is being delivered.
And then, of course, the next step is going into mice. Can you tell us about that?
Yes, so this is a collaboration with Anita Koshi, who was just here and was just traveling back to the US today. Her and I were applying for some funding together to look at some other studies. Anita is an expert on the interaction and progression of Toxoplasma in the brain. She's a neurologist who's also a researcher. And so she stepped in and helped us do the experiments where we infect mice with the parasite and then ask, do they reach the brain and do they still spit this chimerical protein or these fusions in the brain for real? And if we take some slices and do immunofluorescence now in actually infected brains, can we see some of this protein going into the right location inside neurons in real infected brains? And we're able to find some evidence that really happens also in animals.
And to what extent do the neurons take up? Is it 1%, 10% more? Do you have some sense?
So I have to admit that probably off the top of my head, I can't tell you percentage. What we do have in the paper, and that is yet another group that joined in, that's the Swiss group that joined us. I think that they were the last to join us after reviewer comments, where we took animal brains that were infected with the parasites. And so in this case, not using the parasites that are delivering the therapeutic protein, but instead of that, using the parasites that Anita generated, that from the rock tree, they spit the Cre recombinase, which is almost like molecular scissors that cut pieces in the DNA. And those were put in mice that have a Cre recombinase sensitive sensor. So if the parasite go in and they spit these scissors, the cells that they go into turn green. And so we infected the animals, we harvested the brain, and then the Swiss team was able to slice the brains, clarify them, so that you can now create a 3D scanning of the infected brain and show where are all the cells that went green. And you get there a nice visual representation of which are the cells that the parasite interacted with. So this is rock trees again. So not all of these cells have a parasite residing in them, but they've all been interacting with the parasite. And so there you get to see a bit kind of maybe not the percentage, but the magnitude, and also the areas in the brain that the parasite reaches. But again, there we did not have the fusion protein. So What we still don't have is really how many cells in the brain will actually get the fusion protein.
Although there's a section in your paper where you talk about cleared brains and looking at expression, and apparently it goes to many places: the cortex, hippocampus, brainstem, hypothalamus, thalamus. So it's quite widespread, right?
Yes, at least the cells that have interacted with the parasite, that's quite widespread. Yeah.
Right. And so there's still quite a bit of work to do here, right?
Yeah. Yeah, there's loads of different things to do. And again, many of those not by us. So just like what we've done so far sits on the shoulders of others who have evolved genetic manipulation for the parasites, who discovered the different proteins that are secreted from different organelles, who understood which are the cells the parasites interact with in the animal. This still kind of waits for the field to understand, what drives the parasites to certain cells. Can we change that? Can we use ligands that the parasite interact with to attract it to different types of cells now? And most importantly, what are the things that make the parasite virulent? Can we play around with those in a way that still allows blood-brain barrier passage, widespread interactions in the brain, but that prevents the symptoms that the toxoplasma can cause.
I guess it would also be interesting to know if you could take a mouse with a defect known to involve lacking a protein or a misfolded protein, and then deliver it with toxo and see if you can fix it, right?
Exactly. So this is the kind of reviewer request that came back most frequently. And I think the difficulty is because we can't control those things I was just talking about. We can't, for example, flood an animal with the parasite so that it goes everywhere in the brain without causing damage. And so we were not in the position to do that and then change the phenotype of the whole animal yet. So before we reach that situation, we can't really deliver that kind of level of proof of principle.
Right. So in humans, when they get infected, let's say during their teenage years, that infection remains in their brain for as long as they remain alive. And the AIDS epidemic that happened, and I'm sure you're aware of this, but maybe some of our listeners are not, there was a large number of people in France who were dying from encephalitis, and they didn't know why. And it turned out that these were AIDS patients who had acquired Toxoplasma before they acquired AIDS. And then when the AIDS epidemic affected their immune system, of course, the parasite was released from its hold and caused the death of these individuals. And it turns out that that's exactly so. Trimethoprim and sulfamethoxazole was given as a prophylactic almost to anybody who came in suffering from a headache and who was also men who were having sex with men, let's put it that way. And the epidemic was stopped, absolutely stopped. So why can't you use this organism for delivering of your proteins, and then once they're delivered, give the patient trimethoprim and sulfamethoxazole. Just cure the infection.
Yeah, I should say it's it is true that people who are immunocompromised and were previously infected with Toxoplasma are in danger for that reactivation that you were talking about. And the solution for that could be providing things like pyrimethamine, sulfadiazine, in some cases also tovacone, depends. People might develop sulfadiazine, for example, have terrible side effect and is also immunosuppressive. So kind of maybe two things. One I kind of already said is that some of these therapeutics have severe side effects on their own. So, which is why it's not really a great idea to put the parasite into the people and then say, okay, if you have this, if they start creating damage, we just treat you for the parasite. The other issue, and that's maybe the biggest one, is that when these parasites do go dormant, this part of the life cycle we cannot clear at all. And so you're basically giving people an infection for life. And who can predict what's the next thing that people can get that will be in favor of reactivation. Maybe another also important thing to say, each time there is a reactivation, there is an immune response in the brain, which we don't fully understand yet. And so this idea of giving you parasite and then treating you just for the tachyzoid, let's say you even don't have side effects, there is still an immune response. And then you get it again and again and again, because you need to keep getting it to have the delivery continuous. We don't know what the effects of that is either as a field, really. What's the effect of repeating immune response to toxoplasmosis in the brain? So I guess there are kind of three reasons why we won't just rely on the treatment for acute toxoplasmosis as a gateway.
And you probably also want to keep delivering those therapeutic proteins, particularly in a condition like Rett syndrome. And I assume the dormant form would not be producing or would not be delivering protein. So you'd really need an active infection, wouldn't you?
So one of the reasons why we went for GRA16, the specific dense granule protein we used there, is that at the time there was some indirect evidence suggesting that gets secreted even from the bradyzoid form. The thing was, this data was fairly preliminary, and it was from bradyzoids that were formed in vitro. And there is a whole discussion about how comparable are bradyzoids formed in vitro and the really long-lasting brain cysts. And so we don't really know for sure what, like you say, what are the proteins that get secreted from long brain cysts, long-lasting brain cysts. There might be some. So one maybe misconception is that bradyzoids don't do anything. That's also not true. They're not fully dormant. And so there might be some tool there, but I just don't think we know enough yet.
So you've established that you can use toxo to deliver proteins in various cells and also in a live mouse. So is it now the case that you have to focus on understanding what regulates the virulence, or do you still do that along with further developing it as a vector?
Both, yeah. So, firstly, looking at virulence and primarily what we're trying to identify is whether there are things that the parasite doesn't need when we grow them in vitro, but once they get into the tissue, that becomes essential. Things like oxotrophy, for example. And so we're looking at the series of pathways like that and testing them. And one of the authors on the paper, Daniel Walsh, who just graduated, that was his PhD thesis. And the other thing is to kind of try to improve the fusion proteins. So trying to make the fusion part smaller. So instead of putting the whole therapeutic protein on the whole parasite protein, maybe we want to have mostly just the therapeutic protein with just the piece that moves it out of the parasite, which we haven't yet made great progress on. As well as, when we made these parasites, we used all sorts of genetic materials to select for the genetic manipulation and all sort of things that we do in the lab, because we're not planning on using things in the future. And then if you want to really use a genetically modified tool that will at any point in the future will get into people, you have to minimize those things. So these are more kind of like technical optimizations that still need to happen as well.
Well, the identification of what you need to get secreted is really important, right?
Yes. Yeah. And there is a little bit of that in the paper, just showing that, we weren't able to find that minimal sequence ourselves yet.
Wow, it's a lot of work to do. No wonder you need a lot of people.
and a lot of time. We're looking at, if these things, which we all still see quite as a fiction, if this thing, ever moves forward towards non-fiction, we're looking at years of study. And again, it's not going to be only our lab. It will, as it already have been, will necessitate progress from the field in general in a variety of the things we were discussing.
Gee, Dixon, if you were still working, you could use trichinella to deliver proteins to muscle.
That's right. Exactly right. I was fortunate enough to get to know two people very well. One was Elmer Pfefferkorn, and the other was John Boothroyd. And that's all I heard from them were how they were manipulating this organism to do all kinds of wonderful things. And the one that I thought was the most clever with the least amount of effort was the one that Elmer Pfefferkorn developed, where he took a 96 well plate and put cells in each well, and then he infected all the wells. And the wells were, the cells were B cells. And of course, he was looking for the ones that were important for interacting with the parasite, right? The other one he did, and he found out which B cells were that. But his biggest accomplishment, I thought, was he used a temperature-sensitive mutant to vaccinate animals with. And you stick it in their skin and they grow, and then they can't go further because the body temperature is too high, and so they just die. But in the meantime, they deliver the vaccine necessary, the living vaccine in this case, to immunize them against the Toxoplasma infection.
It's funny that you mentioned vaccine. I'll come back to John as well in a second. One of the avenues we're looking at is using a strain used as a vaccine as well as the delivery instead of making the parasite less virulent artificially, piggybacking on a strain that is already known. This is work that we're doing in collaboration with David Smith at the Moreton Institute, and there is a strain called S48, which is also known as Toxovax, and that's a strain that for some reason does not cause brain cysts in large animals. And
really?
David is trying to understand why that is, and at the moment it's used to vaccinate sheep. And there's all sorts of issues with it. The shelf life are not long, and they have to vaccinate every year. So he's trying to understand, what made this strain the way it is to then make it artificially. And there's a student, Sanaya Crouch, who's her PhD is to try to use that strain as to understand how the strain works, so that maybe in the future we can use that as the delivery strain. And just a word about John, who I just saw online very recently because he was nice enough to ask me some questions about my grant in preparation for an interview. Is that my PhD supervisor, who I was mentioning, I went to study how to genetically manipulate Toxoplasma, did her postdoc with John. So in a way, John is my scientific granddad.
How nice. Great.
Dixon, Christina, anything else you'd like to ask?
Not about the paper, but I was thinking, because you mentioned this is a crazy idea and a crazy kind of paper and crazy work, but you did mention that you also do less crazy things in your lab, and maybe just to kind of round up, you could tell us a little bit about the other things that you're trying to achieve.
I'd love to do that. I think at this point, sadly, most of that still is not hugely attractive to. It's just not as crazy, so it doesn't create as much a buzz. But I think everybody kind of knows mitochondria as the powerhouse of the cell, and we all feel like we know what mitochondria do. They're responsible for nutrients and energy, and how they do it, the electron transport chain, moving electrons and pumping protons and generating ATP. And so we're very interested in the mitochondrion of the parasite, because it's eukaryote, has its own mitochondrion. And over the years, we've discovered that, in fact, the parasite mitochondrion is extremely, extremely different from the host one in many different ways. And that allows both some avenues for drug discovery, because there's just the machineries in the mitochondrion of the parasite are sensitive to inhibitors that the host isn't sensitive to. And then kind of also just understanding that mitochondria can work completely different from what our textbooks are telling us at the moment. And so that's a lot of what our lab is doing is on that kind of curiosity-driven discovery on divergent mitochondria biology, and how can that also inform drug discovery.
Yeah, I was hoping I would live long enough to see mitochondria actually be cultured in vitro, so that anybody could then work on the systems.
You'd be interested to know that some people have shown a few different things that kind of go in that direction. So for example, you can isolate mitochondria from some cells. This is on the parasite now, and put them on other cells, and the other cells will take them in.
Oh, nice.
Yeah, and some labs who are genetically modifying mitochondria and putting them back in. So they are former bacteria, so they have some level of their own mind of their own.
That happened a long time ago, apparently, because we really diverged from that along our own path of evolution.
It's exactly that. And in fact, because of that I think most people kind of wrongly assume that mitochondria are the same in all eukaryotes, because they all came from the same one endosymbiotic event. But actually, there are quite fundamental, big, high-level differences that we're now starting to understand.
Cool.
I have one question for you, which is not related to this, but Lilach, what would you have done if you had not been a scientist?
Oh, what a question. So here is a story that maybe listeners will find waking them up. At some point during my career, I'm not going to say when and where, I encountered sabotage in the lab I was working in, and it was extreme. I could not do my work for almost a year. And at that time, I needed to deal with the soul-crushing idea that science is not going to be for me, because I am not managing to do science. And it revealed to me two things. Firstly, that I love science, because I was really depressed by the idea that may not be the career path for me. But the other thing that it did reveal to me is that if I had to think about something else, I was interested to go into laws, and I was thinking about being a judge. That's something. Yeah. So I'm interested in justice, and I was thinking, if I had to start a career again, maybe that would be the direction for me. I am very pleased that issue sorted itself out in the end.
All right. Okay, that is TWiP 244. You can find the show notes at microbe.tv/TWiP. If you have any questions or comments, you can send them to twip@microbe.tv. And if you enjoy these programs, we'd love to have your financial support. Go to microbe.tv/contribute. Our guest today, Lilach Sheiner from the University of Glasgow. Thank you for joining us.
Thank you.
Dixon Despommier is at trichinella.org, thelivingriver.org. Thank you, Dixon.
Thank you, Vincent. And thank you, Christina, for identifying such an interesting presenter. It was delightful.
Christina Naula is at the University of Glasgow. Thank you, Christina.
Thank you. That was a fun evening, as usual. Thank you, Lilach, for coming along. That was really great.
Thanks
and I think
me.
you brought across something that was really, really complicated in a way that I think is really accessible to me, but also to many of our listeners. So I appreciate that very much.
I hope so. Thank you.
I'm Vincent Racaniello. You can find me at microbe.tv. I'd like to thank the American Society for Microbiology for their support of TWiP and Ronald Jenkins for the music. Now, Lilach, I want you to help me close out. Here's what I'm going to do. It's very simple. I'm going to say, You've been listening to This Week in Parasitism. Thanks for joining us. We'll be back soon. And then you say another TWiP is parasitic. Is that, you got that?
Another TWiP is parasitic.
Well, wait till I say the other thing. I'll say it first, okay? You've been listening to This Week in Parasitism. Thanks for joining us. We'll be back soon.
Another TWiP in parasitic.
Outstanding.
Beautiful.
That was perfect. Thank you.